Hello, good afternoon, everyone. So this is just a review class for the test that's going to be happening on Thursday. And don't forget, you also have to complete your online lab quiz, which is 2.5% of your final grade. I see that some of you actually have done that already. So that's good. That's one less thing you have to worry about. But yeah, we're gonna be doing the test one review. There is I posted an announcement on eCentennial that showed you where to get the review package. And if you haven't had a chance to download it yet, let me just put it in the chat box for you. There you
Vincent,
go. Yeah,
I have a question about ROGAM. So I don't know if you can answer it before we start this
sure,
one.
sure. Yeah, let's do that.
Okay. Yeah, so it's in the study guide for the
Number two, yeah.
number two, yes.
Mm-hmm. Yeah.
Uh,
Is it one of the application question?
yes.
Yeah, yeah.
Because I also answered it in the test, the self quiz, but it was wrong. This one, yes.
Which one? Sorry.
The second one, the number two.
Second one? Yeah, okay.
Yes.
So Sally's first child was born with hemolytic disease after newborn. She learned about ROGAM from her friend. Sally is now pregnant with her second child, and she is requesting to receive ROGAM shot from her doctor. Is Sally's request valid? Now, the first of all, like what is ROGAM, right? ROGAM is um, artificial anti-RH antibodies. Okay, so these are made in the lab, and they are the purpose of them is to target. Sorry, my program is a little bit glitchy. I got to restart. Give me 1 second, please. So yeah, it's anti-ROGAM that's given to RH negative mother carrying an RH positive baby, right? Let me just write that out for you. Given to RH negative mother carrying RH positive baby. And the purpose of this is to prevent sensitization, prevents her immune system from being activated by the positive cells, right? In case the baby cells get into the mother's bloodstream during labor, for example, or if there's damage to the placenta. However, Sally was the first child already had hemolytic disease of the newborn. That means she actually got sensitized prior to having her first child. Okay? Maybe she got sensitized through blood transfusion. Okay? So once sensitization has occurred, her body will already make the anti-RH antibody in her system, right? And so there's no point giving ROGAM. ROGAM is not going to be able to undo the sensitization. Right? ROGAM's purpose is to prevent sensitization. So if she already has the anti-RH antibodies in her system, those antibodies they could cross the placenta and attack the baby's blood, right? Uh, if the second child is also RH positive. So the request is not valid. Does that answer your question or you have a follow-up question?
Yes, thank you.
Okay. Yeah, so if you guys have any specific questions you want me to go through, just let me know. Otherwise, I'm just going to be taking up the review package with you, and hopefully it will prepare you for the tests. Okay. So here, the test, by the way, is only going to be lecture 2 to lecture 4, right? That's the test. That's all. 2 to 4. Right? The heart stuff, the cardiovascular system stuff, it's not on. That's in the lab though, right? But not on the test. So here we go. Label the different layers of the fractionated blood in the diagram below and describe components found in each layer. Fractionated blood just means the blood has been separated. We can separate the blood by spinning it at really high speed, right? We call that centrifugation. And the heavier cellular component will sink to the bottom, and then the lighter liquid portion is going to be on the top. So this yellow portion at the top, that is plasma. Okay, plasma. And of course, I'll be posting this up, you know, on eCenteno later. But you know, try to copy whatever you can. What percent of the blood is made up of plasma? Does anybody remember?
55.
Yeah, approximately 55%, right? Very good. 55%. And in terms of composition, the plasma is mostly water with dissolved ions. Right, so there are like different types of ions in the blood. Sodium, chlorine, okay, maybe there's some calcium, so on and so forth. Right? There's also nutrients, wastes. You know, I'm just gonna turn off the camera. You don't need to see me. It's give me more space to write. Wastes and plasma protein. Plasma proteins. Right? We actually learned many different types of plasma proteins. Examples of plasma protein. So can you give me some examples? Anything at all? Any protein that you remember? It's going to be found. Sorry, what was it? No, no plasma proteins. Okay, that's fine.
The fibrinogens.
Yeah, so we have things like clotting factors, like clotting protein that helps you stop the bleeding, right? Things like fibrinogen, and the other one is called prothrombin, right? So these are clotting proteins. Remember, I told you there's going to be questions where you have to reorganize the steps in terms of you know, what activates what. There's the thromboplastin that we talk about, along with the calcium. It activates things one by one, right? Jackie has another one for us. Albumin, that's very good. Albumin, it helps maintain osmotic pressure. Basically keeps the fluid in the blood so that it's not going to be leaking out, and maintain osmotic pressure. What else? Any other proteins? Oops, there's a spelling mistake here. Osmotic pressure. Yeah, antibodies, exactly. So we have immune proteins. Immune proteins, such as antibodies. Right? We also learn about complement proteins. If you don't remember what complement proteins are, that's okay. I'll review that with you as well. Okay, so lots of proteins in the blood, in the plasma portion of the blood. Now, in the middle here, we have what we call a Buffy coat. Okay, Buffy coat has two things in it. Contains the platelets. Platelets, they are cell fragments from megakaryocyte. And of course, they are needed for blood clot. And also they have no nucleus. Okay. Red cells don't have nucleus. Platelets don't have nucleus. Only the white ones do. So along with the platelets, you will also find the white blood cell, WBC, right? Or the leukocyte. This one is thrombocyte. So the Buffy coating is just about 1%. Very, very small. Now, in terms of white cells, we learned 2 different categories, right? For white cells. Does anybody remember what the 2 categories are?
Granule and agranule.
Yeah, exactly. Thank you. Granulocytes and agranulocytes. And granules are these sacs, if you would, that contains enzymes in it, and you know, whether you can see it or not, that's gonna determine whether it's granulocytes or agranulocytes. So granulocytes, they have visible granules. And there are a few that we talk about, like neutrophils, eosinophils, mast cells, and basophil. Now I know, when we took the note, the first time we wrote the note, we didn't put mast cells here, right? That's because we didn't learn mast cells until like lecture 3. But mast cells is similar to basophil, right? They both play a role in inflammation. Okay, the difference is that the mast cells are like stuck to the tissue for like a localized kind of reaction, but basophil, they kind of move around, right? Agranulocytes. We have monocytes, which will become macrophage. Okay, remember, macrophage is also an APC, right? Antigen presenting cell. It's able to show other cells, the naive T cells, what they have phagocytized, what they have eaten up. And you know, we also have the B cells and the T cells. Okay, we'll review what they do later on. There is a question coming up, but I just want to list them here to help you refresh your memory a little bit, so you know, which category they belong to. Okay, how about this last portion here at the bottom? What do we call this bottom section of the blood?
Hemoglobin.
Okay, yeah, I mean it has hemoglobin in the cells, but you're very close. I think you're thinking of the other word. That's also, yeah.
Hematocrit.
That's right, you got it. Yeah, hematocrit. Very good. Okay, hematocrit, right, which is basically makes up the 45% of the total blood volume, and this contains RBCs, right? Red blood cells, and the fancy word for red blood cells is erythrocytes. Erythrocytes. Okay, so that, just some random stuff for red cells, like no nucleus, contains hemoglobin. Hemoglobin is for O2 and CO2 transport, okay? In terms of the shape of the red cell, it is biconcave shape. Why? so that we can increase surface area, right? For gas exchange. How long do they live for? How long is a lifespan of a red cell, huh? Yeah, very good. 120 days. A little longer than 45. Okay, 120 days lifespan, and they will break down at liver and spleen. And also, just like a side note, remember the kidneys can secrete EPO, erythropoietin or EPO, to increase RBC production. All right. That's pretty much it for red cells. Okay. Put a star here, here, and here. The red cell, white cells, and platelets, we collectively call them formed elements. Formed elements. And all these formed elements, they are produced in the bone marrow. Through hematopoiesis. Hematopoiesis. That's a process by which the stem cells in the bone marrow, they are going to become the white cells, red cells, and whatnot, right? And even when they become white cells, they have to decide what kind of white cells they want to become, right? Like, it's a neutrophil, is it a mast cell? Like, you know, there are different options. Okay. Everybody good with this stuff? Anything you want me to clarify before we move on? Just, you can ask anything. No? All right. Compare the two test tubes shown below. Explain what might cause the results on the right. Now, this one is the normal one. Okay, even though, like, I know, like, even though when you look at it, you're like, hey, how come there is not a lot of hematocrit here, right? Okay, I'm not sure. Maybe the person made a bad collection or something like that. Okay, but the one on the right is definitely not good. This one is definitely not looking good. Okay, so can you tell me, like, what might be the problem here? Any suggestions? Why do we see so much red stuff at the top here? What are some possible causes?
Dehydration?
But why would you see all the, like, why would it look bloody in the plasma? If someone is,
Homeostasis.
yeah, that's right. Okay. Hemolysis, I think that's what you meant, right? Hemolysis. Okay, so not hemostasis, right? Hemolysis or hemolysis. This is basically RBC rupture, right? And all the hemoglobin basically leaked out. Leaks out and is in the plasma now, right? Causing the plasma to turn red. And why would someone experience hemolysis? I mean, there are many reasons, but what is What is one reason that we learned in the course?
Blood incompatibility during transfusion.
Very good. Mismatch. Blood transfusion. Right. Incompatibility, like you said. Or maybe internal bleeding. Internal bleeding. Okay. That's good. Remember this for the test. Right. There's going to be a question like that. All right, matching up babies. There will also be a matching baby question on the test. You should have plenty of practice by now, but you know, you just have to remember the 4 rules, right? Let me write down for you again. AB cannot have O. Of course, like I'm like condensing it now, right? Like for the full thing, you can look back in the notes. O parent cannot have AB kids. O plus O equals O. Whole family is going to be O. RH negative and RH negative is going to be whole family RH negative, right? So if you remember these things, it's going to make it easier for you to do the matching question. Okay, you shouldn't be doing Punnett squares for these type of questions. It's not a very good use of time. Okay, so let's start with parents that have AB or have O, and even better if they're, you know, one is AB, the other is O, right? Because that's the combination where we can eliminate a lot of babies, right? So if the one parent, I don't know if it's the mom or dad, it doesn't even matter. If one parent is AB, then automatically I can cross out baby I. Okay, no O, right? The other parent is O, so no AB. So which one is it? Is it baby J or baby K? How do you know?
Baby K.
Why? How do you know?
They are RH negative.
Yeah, both of them are negative, right? So the baby has to be negative. So this is it. Baby K, done. Okay, so baby K is gone. We can choose another parent that has O in it. Let's just do this guy. So no AB, and they are both negative, so you know, no positive baby. So this is baby I. And besides, it's just A and O in the parents. They will never be able to produce a type B baby, right? So that's that. Baby I is gone. Looking at this couple here, there's an O, so H is not there. So this is J. O cannot have AB, right? And then that's it. Okay. So yeah, there will definitely be a baby matching question. Make sure you know how to do them. All right, this next part here is like confusing for many people, but let's try to do it. Okay, a new blood type called Fluffy has been discovered in a human. Like I just make this up. This is not real. Okay, there are. Sorry, I have a question in the chat. 2 negative can have positive baby. Nope. 2 negatives cannot have positive baby, but 2 positive can have a negative baby if they are heterozygous. If it's like this, right? Then there's a 25% chance that they can have a negative baby. Does that answer your question? Okay, yeah. Negative, you can only have negative, right? Because there's only one way to be negative, right? Like that. 100% will be negative. Okay, Fluffy. A new blood type called Fluffy has been discovered in humans. There are 3 typo. 3 alleles in the Fluffy blood group, F1, F2, and F3. Okay, and here are the rules. If F1 is dominant over F2 and F3, and then F2 is dominant over F3. So if you have to write out, it looks like this. F1 is dominant, bigger than F2, which is like bigger than F3. It's going to cover it up. That's what I mean. How many phenotypes are there in the fluffy blood group? Now, before we can do the phenotype, we have to do the genotype first. Genotype is the genetic makeup. Okay, remember, it doesn't matter there are many alleles in the population. At most, we can only keep 2, right? Because humans are diploid. At most, we can only keep 2 alleles. So there are 3 alleles in the population. What are the possible combinations for people? Well, it could be 1-1, like F1, F1, 2-2, 3-3. Give me another combination, please.
1, 2,
1 and 2.
Maybe another one.
2, 3.
Yep, and one more.
Okay.
Yeah,
And 3 and 1.
exactly right. Okay, so those are the only possible combinations. There are a total of 6 combinations. Okay, it's just like ABO blood group, right? You have 3 alleles in ABO. IA, IB lead to I. There are 6 combinations. So now let's take a look at the phenotype. Okay, the phenotype is the expression, right, of this thing, right? So let's see. If the person has 1-1, then his blood type or her blood type is going to be, we'll call it like fluffy 1. Okay, fluffy 1. Again, it doesn't matter what you call it. I'm just making this up anyways. Do we have another fluffy 1 here? Is 2-2 fluffy 1? Like, I mean, obviously not, right? Because there's no 1 in there. Yeah, but is there another one that's fluffy 1?
1, 2.
Yes, because it says 1 is dominant over 2, right? So when you have 1 and 2, only the 1 is going to show up. 1-3 is going to be also fluffy 1. Okay, because 1 is dominant over 3, and it's dominant over 2. So we can choose another one here. This one we will call fluffy 2. Do we have another fluffy 2, or that is it?
2, 3.
2-3 is also fluffy 2. That's right, because 2 is dominant over 3. When you have 2 and 3, the 2 will cover up the 3. Only the 2 will show up. And finally, we have 3-3, which would be fluffy 3. Okay, so there are going to be 3. The answer is 3. All right, let's take a look at another scenario. Same thing, still talking about fluffy, but it's telling you that they are all codominant with each other. Codominant with each other. Okay, so the genotype does not change. It's still the same 3 alleles. Therefore, we have the same combinations. Now, let's talk about the phenotype. Phenotypes. How many phenotypes do we have? I am going to ask you to do a poll. You tell me which one it is. Okay. You can go not sure if you're not sure. Okay, thank you. Yeah, I mean, the answer is is actually 6 here. Okay, the answer is 6. Why? Well, 1, 1 is going to be fluffy 1. I hope that's easy to understand. 2, 2 will be fluffy 2, fluffy 3. That's not a problem. What about this one? 1, 2. Well, the question says they are codominant with each other. Codominant means both allele are going to show up. Right? So it's not just 1, it's not just 2, it's 1, 2. Right? To connect to what we learned, when the person has IA, IB, the blood type is not A, the blood type is not B, the blood type is AB, right? 2 things at the same time. So that's its own blood type. 2, 3 is a blood type, and 1, 3 is a blood type. So therefore, there's 6 different alleles. 6 different phenotypes. Is that okay? This is going to be on the test. So please let me know if you need me to explain further. Okay, very good. Find the genotype of question mark. So we have to figure out the blood type of these people. And if you see a pedigree question like this on test, we start with type O and type AB. Those are the people that we know about the genotype. There is no question about it. IAIB, little I, another little I here. Right? Those are the only ones we know for certain. Like off the top of our head, right? Then for everyone else, we can write half of it. Right? It's going to be IB something and IB something. Over here is IA something, IB something. That's all we can do as a first step. And we sort of have to do some like detective work. Right? We have to think about, okay, where does this little I came from? Well, one of the little I must be from the mother, and then the other little I must be from the dad. Okay. And then and then if we take a look at the other kids, like this IA, where does the IA come from? Well, IA can be from dad or from mom, but if we trace back another generation, we see that like the father side, nobody has an A, right? So it's not possible to get the A from there. It must be from the mother side. Right? So the mother in this situation must be type A. Okay, but they're looking for the genotype, so like heterozygous, right? And for the other one, this IB must have came from the father side. But that's as much information as we can get. There's no way to tell what these are for certain. I mean, both of them could be IB little I, or like maybe only one of them is IB little I. I don't even know which one is the little I inherited from, right? But that's okay. That's not what the question is asking, right? This is as much detail as we can get. I mean, we can also figure out these are heterozygous, but that's all. Okay. Here we go. A drop of Sarah's blood was mixed with anti-A antibodies. Okay, remember in the lab, there is like, or like even in the lecture, right? There are these plates that has A and B and Rh antibodies in them. So they're saying a drop of Sarah's blood was mixed with anti-A antibody, and another drop was mixed with anti-B, and then anti-Rh. Clumping was not seen in any of the tests. So no reaction, no reaction, no reaction. What must be the reason? Well, let's take a look. A, the test must have been defective because clumping shouldn't have been seen in at least one test. That's not true. Okay? It's totally valid to have no clumpings in any one of them. Okay. None of the antibodies in Sierra's plasma reacted with the antibodies. That's not true. It should have said none of the RBC antigen reacted with the antibodies. Okay. The antibodies are already in the plate. We're trying to see if they will bind to the red cell. Sierra's blood type is O positive. What do you guys think? Do you think it's O positive?
No.
No.
No, it's
Yeah.
O negative.
That's right. O negative, exactly. Because there's no reaction with RH. How can she be positive? So Sierra does not have any ABO or RH antigens on her RBC. That's the answer. That's why there's no reaction. She doesn't have these things. There's no A, no B, no RH.
Vincent, isn't O part of the ABO?
O is part of the ABO, but O is the absence of A and B. Right? There's no such thing as like an O protein. O is missing A and B, right?
Yeah, but the option says Sarah does not have any ABO.
Oh, I see what you mean. Yeah. So, I mean, like you just refer to them as like a group, right? So no A, no B. Basically, that's what this means. Yeah, I see what you were saying, but that's how we refer to them as. Okay, even though she's type O. I know, it's a little bit confusing. Valid point though. All right, number 7. Okay, some of you were having trouble with this. Hopefully, it will make more sense now. Okay, very good. You know, for blood transfusion, just have this with you if you are still having a little bit of trouble figuring out who's safe and not safe. You know, this is gonna help you just visually see who can give to who. So positive cannot be given to negative. You're gonna sensitize them. We don't want that. Unless it's an emergency, right? So A and E, it's gone. And then the rest are actually okay, right? All of these are safe. All of these are safe. Does anybody want me to explain like why they are safe, or is there a particular one that you're not sure why it is the way it is? And you need me to explain, just tell me which one. And I'll go through it. Okay, so expect some transfusion questions on the test. Okay, fill in the following chart. Macrophage. So classification. How do we classify macrophage? I mean, there are more than one correct answer here, right? Macrophage. Anybody like, you know, off the top of your head, If I say macrophage, what category comes to your mind?
Agranulocytes.
Same.
Sure, yeah, they are agranulocytes. So that's like morphology, right? Morphological classification. You classify it based on how it looks. What about functionally? What do we classify them as based on their function?
Phagocytes.
Yes, they are also phagocytes. And also they are APC. Okay. So yeah, not all phagocytes are APC. Okay. Neutrophil is a phagocyte, but it's not an APC. What is the main function? They recognize PAMs, which are pathogen-associated molecular patterns, so that they know what to eat, right? What belongs to the body and what does not, right? They are there to do phagocytosis. And they present antigens to naive T cells. Okay, they are the link between innate and adaptive immunity. Right, we talked about the HIV virus. They infect and kill the macrophages. And without the macrophage, the person has no way to activate adaptive immunity. That's why it causes immunodeficiency. Right. Okay, neutrophils. I'm just gonna switch between two colors so that they're not like too crowded. Neutrophils are also phagocytes. They are granulocytes. They do phagocytosis. It is the most abundant WBC. You have a lot of them. That's what it means, most abundant. 70% of your white cells are neutrophils. Mast cells and basophils, they are granulocytes. And what do they trigger?
Inflammation.
Very good. They trigger inflammation by secreting histamine, right? The difference is that mast cells, they are local, whereas basophil, they are more systemic. Systemic means all over the body, right?
Vincent, so local means at a particular site of the injury, right?
Yeah, like you cut your finger, it's only inflamed at the finger, right?
Okay.
Yeah. Localized, maybe. Okay, NK. NK stands for natural killer cell. And I would classify them as part of innate immunity. Okay. I mean, all these things are kind of innate immunity as well. And they do two things. They kill infected cells And they also kill tumor cells. Helper T cells, it's part of adaptive immunity. These are immunity that you have to get throughout life. They evolve, they change. They are able to create tailored solutions for, different type of infections. Whereas innate is like, more non-specific, right? More general. If you remember the note we took, helper T cell, like anything that deals with T cells in general, we call it cell-mediated. Cell-mediated immunity. What does helper T cell do? They activate naive B cell. Right? They have to find the B cell that has the right antibodies so that they could clone them, right? In general, they coordinate immune response. Plasma cells. Plasma cells, they are part of, and also part of adaptive immunity. Adaptive immunity has two branches, right? The T cell branch and the B cell branch. The B cell branch is what we call the antibody mediated immunity. And there's only one job for the plasma cell. And what is that one job? What do they do?
Produce antibodies.
Yeah, that's it. They mass produce antibodies. Now, while we have a moment, let's talk about what antibodies do. Remember, antibodies, they are like Y-shaped proteins. The arms, they are the variable region. And then here we have the constant region. The constant region, as the name suggests, is the same for all antibodies. But the arms, variable regions, are going to be different. They're going to be unique every time you make them. Oh, there's a question. What does cell-mediated immunity means? I mean, like, so like that's just a name of what they're called. But more like if I have to give you a reason for why is it called cell-mediated, it's because the branch, the T cell branch, the immunity that's created by the T cells are mostly carried out by cellular action. So like the helper T cell is going to go activate something, right? The killer T cells are actually going to go kill other cells. So like a lot of the actions are carried out by the cells. Whereas for the B cells, it's not the plasma cells that's going to be going around attacking things. Okay, it's what's produced by the plasma cells that's doing the attacking. That's why this is called antibody-mediated immunity. Right? So whether it's cell-mediated or antibody-mediated, it's describing the active ingredient, if you would. Okay, is it the cell that's attacking or is it the antibody that's attacking? That's what the name difference is, okay? So antibodies that you create for one thing is not going to work for something else. Generally, it doesn't. Sometimes we get lucky, right? Like the case of tuberculosis I was telling you. You can, get vaccinated with the cow version, and you will get protected for the real one, from the human one as well. Generally, how do antibodies work? Well, they neutralize. This is too thick. Let me tone it down a size. Neutralize toxins and viruses. They can also form immune complex. These are all from the notes, okay? I'm just writing it for you. Immune complexes are like, when the antibody binds to their target, just makes them clump together. It's bigger. It's easier for white cells to eat them up. That's all. And they can also help activate complement proteins. If you've forgotten what complements are, I think there's a question coming up. We'll talk about it then. Do I have a question coming up? Okay, maybe not. Okay, so let me just write it down for you as well. Okay, complement. See, there's so many things, like layers within layers, right? Complement proteins is part of innate immunity. Okay, innate. You're born with the ability to activate these complement proteins, and they do three things. They can leave chemical trails for white cells to follow. They can tag pathogens. Remember that Finding Waldo example I give you? Like trying to look for that cartoon character in the picture. But if it's tagged, it's easier to see. And then they can also form membrane attack complexes. Okay. So sometimes, you might find it helpful to do mind maps when you're studying these things, right? So like on a piece of paper, start with immunity in the middle, and then just start writing other big concepts. Okay, so for example, immunity, you can think of like innate, and then from that you can think of other stuff, right? Like fever, phagocytosis, physical barrier, right? And then you can go like adaptive, right? And same thing, so on and so forth. Sometimes you create these mind maps. It's going to help you link the different concepts together. All right. All right, so this is not an exhaustive list. There are other cells you can add yourself, like suppressor T cell, cytotoxic T cell. There's memory T cells, B cells, right? A lot of other things you can add. Okay. That's it. Some more multiple choice questions here. How would you contrast cytotoxic T cell is not NK cells, okay? Cytotoxic T cell is also called killer T cell, but killer T cell is not same as natural killer. Natural killer is part of innate. You're born with it. Okay? Cytotoxic is specific. Okay? You have to create them. So these cytotoxic T cells that you create are designed to kill a particular type of pathogens if they infect your cells. How would you contrast specific and non-specific defense? Okay, so just very briefly on the side here, specific is adaptive. Okay, it's long-term. It's not immediate. Right, and it's specific. Specific means only like one target. Non-specific is innate. No memory. usually works right away. And it's wide range, more than one target. But like, you know, general stuff. Things are not terribly dangerous, okay? Like talking about mostly harmless things that will get you sick, but like not gonna kill you, right? All right, let's see. Specific immunity works only after non-specific have failed. Doesn't have to be, okay? Can be parallel. Okay, and a lot of times they work with each other, right? Like the antibodies will go back and help with the complement, and then the complement can help activate as well, the adaptive as well. So it's not one after the other. Things in the body don't normally happen, you know, in a specific order necessarily. Non-specific defense and specific defense require prior exposure to be effective. Nope. For non-specific defense, no prior exposure required. Next, B cells work in both non-specific and specific defense. Nope. It only works in specific. It's part of the antibody-mediated immunity, right? This is also wrong. T cells also only work in specific. So this is the correct one. Specific immunity changes throughout a person's lifetime because you get exposed to different things. Every time you get exposed to a new pathogens and you recover from it, you gain lifetime protection from it. Non-specific defense does not change though. Fever is going to be the same in a baby as it is in an older person. Okay, complements are going to be the same. So the answer is E. Next, which of the following statements describe a form of acquired immunity? So we did that chart, right? Let me see, where is it right now? Is it this one? Maybe. Yeah, we did this chart, right? So make sure you go through it yourself. There are quite a few questions on this stuff where you have to figure out, you know, what's natural passive, what's artificial passive, so on and so forth. I think this is pretty self-explanatory. You should be able to understand this by reading the flowchart. But, you know, if you look at it and you're still lost, just email me. Here are some examples. A RhoGAM injection is an example of artificial active immunity. RhoGAM is not given for the purpose of immunity, right? And we're not giving you RhoGAM so that you don't get sick from a pathogen. RhoGAM is an intervention for sensitization. So anything with RhoGAM here is like, you know, not the answer. Nursing an infant to provide the antibodies in breast milk is an example of natural active immunity. That's not true. It's natural passive. When you pass the antibodies through breast milk, the antibodies are produced by the mother, but not the baby, right? So the baby is just receiving it. That's why it's passive. It's not doing anything, the baby. It doesn't have to make its own antibodies, right? That's why from the baby's perspective, it's passive immunity. Catching COVID-19 and recovering from it should give you natural active immunity. That's correct. Okay, natural, because you catch the disease, and then active means you have to make the antibodies yourself, and you should be protected from that particular strain of COVID, you know, for life. But if it changes, if it mutates, then, you know, you need a different kind of antibodies. So that's why you will get sick again from another strain. Receiving an mRNA COVID vaccine is an example of artificial passive immunity. And when you receive vaccination, it's going to be active. Okay, that's why that's why when you get the flu shot, when you get the COVID shot, you still, you feel the muscle ache, you get a fever maybe, right? Because after all, you are introducing foreign material into the body, right? That fever, all that, if you feel a little bit unwell for 1 or 2 days, that's your body ramping up the active immunity. That's your body trying to find the naive T cells to clonal, expand them, and produce antibodies and so on and so forth. Okay. So to recap, if you make the antibody yourself, then it's active. If something else makes it and you're just receiving it, if someone else makes it and you're just receiving the antibody, then it's passive. Artificial means there's some kind of injection involved. Natural, no injection. All right, name the cell. Uses porphyrin to put holes in infected cells. I don't remember if I told you about porphyrin. It's just a protein that makes holes. And the answer here is a cytotoxic T cell, aka killer T cell, not to be confused with natural killer cell. Releases histamine to initiate local inflammatory response. That would be mast cell. A type of APC that activates naive T cell. That's macrophage, right? You only learn one type of APC from me, so that should be straightforward. It's just macrophage. What's gonna kill cancer cells? Anybody remember?
NK cells.
Yep, NK cells. Forms blood clots. That would be platelets. Even though they're not really cells, they are more like cell fragments, but you need them to form blood clot. Neutrophil is the most abundant. WBC. All right. Give example of each of the following. Formed elements. We have RBC, WBC, and platelets. Okay. You can write the full name if you want. Erythrocytes, leukocytes, thrombocytes. But those are the formed elements. We listed granulocytes already, but here we go again. Neutrophils. They are most abundant. They are phagocytes. Go around and eat things up. Basophils and mast cells. They are related. They both cause inflammation. Mast cell is local. Basophils all over the body. Eosinophil is another guy. I think I didn't tell you what it does yet, but they kill parasites. Two types of lymphocytes, T cells, B cells. But of course, like the T cells can become different effectors, right? They could become like killer T cell, memory T cell, suppressor T cell, helper T cell. We drew a whole note on that, right? Like a beautiful map for that. Coagulation proteins, fibrinogen, prothrombin. Okay, please remember the pathway. Okay, like what activates what? There will be a question like that. Sorry, you're very far away. One more time, please.
The thromboplastin.
What about the thromboplastin?
It's not a coagulation protein, sorry.
Oh, sorry, yeah. Thromboplastin is usually hidden away, right? It's not in the plasma, okay? It's like hidden away underneath the tissue. It's only when you have a cut or something, then the tissue gets damaged, then you have the thromboplastin that comes out, right? Which then, activates these prothrombin and stuff like that. Okay.
Thank you.
Autoimmune diseases, anybody have anything they remember from lecture?
The Graves' disease?
Yep, Graves' disease. The one where the thyroid is constantly turned on by antibodies, your eyes is popping out a little bit, metabolism goes uncontrolled. You can name any, okay? Like another one is like multiple sclerosis. Okay, rheumatoid arthritis. We have a few. I wouldn't give you something that you haven't heard of, okay? It's basic stuff. Physical barrier, we have like intact skin. We have the normal flora, which is microorganism that live on us and in us. Remember, I keep on saying they take up so much space that there are no more room for the bad ones, right? Mucous membrane. Right? Lots of options. Immunodeficiency, we only learned two types. Two examples, I should say. Two examples of immunodeficiency. What are they?
Primary and secondary?
Yeah, very good. And as an example of primary is SCID, the bubble boy. And then secondary is AIDS, right? Acquired immunodeficiency.
Vincent, in the notes, you said HIV, like causes AIDS.
Yep.
Okay, so is HIV like the
virus?
The virus, okay.
Yeah, HIV is human immunodeficiency virus. When a person gets infected with HIV, the virus kills the macrophages, it cripples the adaptive immunity, then the person has no immunity. Now the person has AIDS. Okay, it's possible for people to be HIV positive, but not have AIDS. Okay, it's possible to have drugs to kind of like suppress the virus enough so that it doesn't affect the immunity, but they still have the virus. HIV positive. Lymphatic organs, spleen, tonsils. There are three pairs of tonsils that we learned. Adenoid, lingual, palatine. It's all in the notes. Okay, here I'll write out for you. That's the one at the back of the nose. That's the one at the back of the tongue. And then there is the one at the back of the throat. Can I have another lymphatic organ, please?
Thymus.
Yes, there's the thymus. That's where the T cells are going to mature, right? Example of artificial passive immunity. Artificial means you have to inject something. Passive means you didn't make the antibodies. So example would be like a serum. I remember I got bitten by the dog and I have to go get the serum in case I catch rabies. Natural passive. What's the example that we learned for natural passive?
Breastfeeding.
Breast milk.
Yep, breastfeeding. Doesn't have to be breastfeeding. I mean, the baby can be inside the mother still, and the antibodies can transfer through the placenta. Characteristics of inflammation: redness, swelling, pain, warmth. Right, and these are the side effects of histamine, right? Okay, just a couple more. Why do we need to receive a flu shot each year in order to have protection against the flu virus? So I told you there are different kinds of flu viruses out there, right? The H and the N antigen on the surface, there are many different kinds. So we don't know which one are going to be circulating each year. That's why you need to have a different vaccine each year, because the dominant strains, the top 3 strains that are going to be circulating is going to be different. Okay. We actually did this one as a poll question in class. I'll just circle the answer for you. You can check the recordings for the explanation. Okay, so there's this graph for breathing. Let's just label the stuff again to help you recall the information. If it's just going up and down like this bit of air that cycles in and out of your lungs, it's called the tidal volume. Tidal volume. So like when you exercise, tidal volume is going to go up. Okay, because you're breathing deeper, right? C here is the inspiratory reserve volume. It's the air that you can breathe in beyond your normal inhalation. So like this is 6,000. This is around 3,000. So the inspiratory reserve volume here is like, 3,000 milliliter. I mean, it's going to be different for different people, right? It's going to be different for different graphs. So you have to, figure out the numbers. For example, here it asks, what is the expiratory reserve volume? Okay. This here is the expiratory reserve volume. Right there. And I mean, this is not the perfect graph. I keep on saying this every time we do a review, but then I never really got around to changing the graph. I really should do that maybe during the summer. Anyways, if I give you something like this on the test, it will be very clear, right? Let's say, so this is, I don't know, that's like 2,400 maybe. It's not quite halfway, roughly 2,400. This is around, let's say 1,200 maybe, 1,200-ish. So if you do the subtraction, it'll be around 1,200. Okay. The numbers are not like the key thing here. The key thing is like, are you able to tell that this is the expiratory reserve volume, right? What is the label for B? B is this whole thing. Whole thing. So we have the IRV, the tidal volume, and the ERV. If we add up all of them together, it is called vital capacity. Okay, so your vital capacity does not change. Right? So listen carefully, huh? When you are exercising, Here, I'll write down. During exercise. Like I'm talking about like running, for example, okay? If you're running, you will increase your tidal volume. This middle portion is going to go up, but then you will decrease your IRV and you will decrease your ERV. Okay? But vital capacity, unchanged. Total does not change. But like how you allocate the available volume is going to change depending on what you are doing, right? Hey, describe the breathing pattern. I'll just quickly do that for you. Okay, so this is just breathing in and out normally. Nothing drastic, nothing different. And then you breathe out as much as you can. Breathe out as much as you can. All right. So at this point, the lung is at its lowest volume. But it doesn't mean you have no air in the lung. You will always have some air in the lung unless it pops. What do we call this air that's like not coming out? Anybody remember that?
A dead air?
Yeah, the dead air. Everybody remember the easy name, right? The dead air. But it's the residual volume. Okay, dead air. It's dead air because it is very high in CO2. It does not participate in gas exchange. Okay, does not participate in gas exchange. It's just kind of like sitting there, like taking up space and doing nothing. Okay. So then the person will breathe in again and then just normally, and then they will inhale as much as they can. Okay. That's pretty much it. You can describe the rest yourself. Done. Draw a flowchart to illustrate how inhalation and exhalation occurs. I mean, we did that just last week, right? So I'm not gonna do it again, but I'll copy the picture from before. It's basically this stuff that we talk about, right? How when you breathe in, your chest has to go up, your diaphragm has to go down. And because of the pleura, because of the membrane that surrounds the lungs, your chest movement and your diaphragm movement will hold the lungs with them. Okay, so collectively they increase the chest volume, which means the lung pressure is going to go down. And if the pressure in the lungs is lower than the atmosphere, then the air comes in, right? Okay, so this is missing an arrow, but then air is going to go in. Right? So, review that yourself. We just did it last week. There's no reason to go through it again. Next one, during swallowing, which of the following happens? The larynx moves upward against the epiglottis. Remember, the epiglottis is the elastic flap, elastic cartilage that covers the glottis. Glottis is the opening to the larynx. And, again, so many layers. And this is something you should practice doing yourself, right? And the larynx is where the vocal cords are, right? Okay, it's protected by the thyroid cartilage and so on and so forth. So if you put your hand in the throat and you swallow, you will feel the larynx pushes up against the epiglottis. That's the answer. The epiglottis itself does not move. That's why that's not the answer. Okay, pharynx is the throat, like further up, has nothing to, it's like before the epiglottis. And trachea is further down. The answer is A. Surfactant. Surfactants are produced by septal cells We also took a note on this. And the job of the surfactant is that it breaks surface tension. It will increase compliance. Compliance is the expandability of lungs. If there's too much surface tension, then it's difficult to open up the lungs, like those babies that are born prematurely I told you about, right? So, surfactant in the alveoli helps decrease surface tension to allow inhalation. Last but not least, the urge to breathe is primarily driven by increase in carbon dioxide. Sorry, wrong one. Increase in carbon dioxide in the blood. This is detected by the respiratory center. Located in the medulla oblongata. Right? And that's part of the brainstem. Okay. That is it for the review. I mean, most of if you know everything on this review package, you're pretty safe, I think. Okay, but like if you want to like the high 90s or like mid 90s, then you might have to do a little bit more studying on your own, with the application questions and stuff like that. But in terms of general knowledge goes, that's pretty much it. Anything else you guys want to ask before we end?
When you get the chance, could you please post the class notes from last week?
Did I not do that? Oh my goodness. Sorry. I will do that right away.
Thank you.
Yeah. Hold on, let me see where is that class note. Last week, no, I think I did, didn't I? It was oh sorry, it's 21st and 21st and 23rd. Oh no, I did not. Yeah, you're right. It was the 30th, right? I think maybe. Okay, I'll do that. Okay, that's it. That's it for my section. Actually, I won't be seeing you guys again. I'll be here. Right? I'll be here for another week next week. I'll be at the college. Okay, but like Miran's gonna take over starting the 11th. No class on Thursday. You just do the test and that's it. But if you need to reach out to me for other things, I'll be here until the 14th. Wish you all the best for the rest of the class. Rest of the course, I mean. And yeah, stay in touch. If you need a reference in the future, feel free to contact me. Yeah, that's it. Okay, take care, guys. Bye-bye.
Thank you. Thank you.
Thank
you.
Professor.
No
Thank you
problem.
very much.
You guys are
And
so
thank
nice.
you for help, Professor.
Okay, thank you.
Thank you.
Bye.
Bye.
Bye, Professor.
Bye, Professor.
Bye.